Morning, everyone. I think we'll go ahead and get started. And it's really an honor and a pleasure this morning to welcome Charlene Zettel, who is one of the UC Regents. But she really is what I would call a Renaissance woman. Some of you may know she served as an assemblywoman in the California legislature for several years. And she's also been CEO of Donate Life California. And if you're not familiar with that organization, that is the organization that maintains a registry of transplant organs, which are really valuable, obviously, to people with life-threatening chronic diseases. And I understand last night she retired as CEO, so now she has all this free time to come and talk to us this morning. Charlene.
Good morning. Well, thank you very much for a lovely introduction. And I'd love to say I have all this free time, but I'm still going to be working over the weekend to close out the office. But as a regent of the University of California and longtime local resident, it's really my pleasure to welcome everyone to San Diego, La Jolla. And as many of you know, we locals call our city very humbly America's finest city. But despite today's cloudy skies, San Diego is known for a perfect climate for research in life sciences, biotechnology, and pharmaceutical development. And in researching this morning for welcoming remarks, it was remarkable to realize that former President Clinton recognized our cluster here as one of the finest genomic research centers in America, calling it the research capital in America of genomic research. So that's high praise and recognition for this area, and you can understand why I'm so very proud of the facilities, the university here at the University of California. Many of you may not know that as a regent, I am privileged to govern 10 university campuses, 5 medical centers throughout California, but UC San Diego is definitely my favorite, and probably because I've watched it grow to international prominence for most of its 55 years. And of course, one of the jewels of the UC San Diego is the Skaggs School of Pharmacy. The Regents approved the school about 15 years ago. And in that very brief span of time, our founder, Palmer Taylor, his vision and leadership has propelled Skaggs to international recognition, of course. The Skaggs School of Pharmacy and its graduates are recognized as some of the best in the United States, according to the U.S. News rankings and the quality measurements of its graduates. As you know, the ultimate purpose of a research university is to convene the brightest minds to collaborate, research, and translate their discoveries into technologies, products, and services for the betterment of mankind. In order to provide for the public good and service, one of the guiding goals of the University of California, and in particular UCSD and the Skaggs School of Pharmacy, is to serve as leaders in global learning, research, discovery, and innovation. The symposium today reflects that sense of service. Building upon the vision of Skaggs founder Dr. Palmer Taylor, the symposium has been designed to provide a stimulating, nurturing, and collaborative environment where men and women of science can come together to challenge each other, expand their research horizons, network with colleagues, and learn. Today you have the opportunity to collaborate with and exchange knowledge with other cutting-edge researchers convened here. Researchers whose discoveries and therapeutics are poised to conquer diseases that previous generations believed were incurable. Cancer, Alzheimer's, and Parkinson's, to name a few. The knowledge you bring here from your own work at universities and research institutions, deepened and expanded by the collaborative learning that will take place here today, will ultimately enable community pharmacists to better ensure the health and safety of the patients they serve. And by solidifying and strengthening pharmacists' role as healthcare leaders and key decision makers in patient care, your brainstorming, learning, and work here today has the power to save thousands of lives and ultimately improve the quality of life for millions more. I know that you're faced with many, many challenges in your careers here. For instance, changing demographics with over 76 million aging baby boomers who are and will be facing significant health issues and are being prescribed a plethora of medications. Potential adverse drug interactions in patients with multiple prescribing specialists, increased consumer demand for the latest miracle drugs, personalized therapies, and emerging technologies, the complexity and impact of the Affordable Care Act, the shrinking healthcare dollar, and finally, cuts in state funding and federal research dollars, which couldn't be more ill-timed or ill-advised. As a regent and as a consumer who is one of those 76 million baby boomers, I understand the critical nature of the work you do. And I believe it's essential that I, as a regent, encourage and educate others in public policy so that we can better understand your complex healthcare environment, understand and appreciate the leadership role of the pharmacist in patient care, work collaboratively to create a regulatory environment at the federal and state level that enables your research to thrive, and to demand the appropriate resources to help your work flourish. In your hands, the lives and well-being of not only millions of Americans, but multiple millions of people from around the world are made better. Simply put, you are our hope and our future. I truly thank you for all you do. As a regent, I'm grateful knowing that because of you, the patients at our 5 academic health centers will increasingly have the finest care that science and healthcare professionals can provide. As CEO of Donate Life California, the state's organ and tissue donor registry, I'm grateful that because of you, the hundreds of transplant recipients I've come to know and love as family and friends are going to live longer lives because their nephrotoxic anti-rejection medications are a thing of the past. In closing, thanks to all of you for coming to the Palmer-Taylor Research Symposium, and special thanks to Dean McCarroll, the conference chairs, and the entire staff here at the Skaggs School of Pharmacy for convening all this brain power in one beautiful setting. So thank you for taking the time to come and share your knowledge and experience. Thank you for inviting me to join you. Have another great day.
Okay, we have a wonderful group of speakers this morning, and I'm going to call up the first speaker, who's an old friend of UCSD, Susan Amara, who received her PhD here some years ago. I actually was on her thesis committee, so I remember that well, and she's had a stellar career and is now a scientific director of the National Institute of Mental Health. Susan is going to talk on neurotransmitter transporters and the cellular action of amphetamines, a walk on the presynaptic side. Susan, we welcome you back.
Thank you. Thank you. It's really a pleasure to be here, although it's a little bit daunting because it's like coming home and giving talks to your parents. And so I wanted to say a couple of things about my interactions with Palmer, and I'll tell you one little story because I think we all get a chance to do that at events like this. So when I was a graduate student, I both rotated in your laboratory. You probably don't remember this, and I also took my first reading rotation. I think it was my second semester, my very first semester. And I had always wanted to be a pharmacologist. Since I was in 8th grade, I went to a drug company and I saw you could have a job and do experiments, and I thought this was great. And so when I came, I did a reading rotation with Palmer, and it was on the effects of sodium ions on the conformation of receptors and how sodium ions altered receptor binding. I remember this very explicitly, and I was absolutely intimidated because I'd never had a one-on-one sort of relationship with a real scientist. And so I would read these papers, and then I would go in, and I would be so nervous that I would forget what I had read. But what I loved about Palmer, and I think you'll recognize this in him, was that he was so patient. And he would just simply ask a question very, very nicely and in a very unfrightening way, and eventually get me to the point where I could talk about what I might know, have learned from that particular paper. But it came at such an important and formative time that it really had an impact on me over the years. And in fact, it was great that you were so wonderful at that point, and I really enjoyed that experience with you. And in addition, I rotated in his lab, and I found it really interesting. But what I was doing was I was doing bungarotoxin binding assays, and I was working with Steve Sine and Greg Wieland, and I realized that they were so focused on this one assay, and I have a fairly short attention span, and I like doing lots of different things, and so I realized I could never be that way. I could not focus in that same direction. And if you know anything about my career, I haven't completely focused. But at the same time, it was a great experience, and I even have a slide which I'll show you. I came back to that later in my career. But so it was a great experience. I only have one picture with Palmer, and I think this illustrates another one of his great strengths, is that this is at the ASPET meeting in 2009, and I have to say UCSD really cleaned up on the awards. And so there's Joan Brown, Palmer Taylor won the Axelrod Award, and Paul won an award too, and only Ken Jacobson was outside of UCSD.
No, he got his PhD in chemistry here.
Oh, okay. So even he's from UCSD. So it was really a good year. I actually, you'll notice I'm empty-handed, and the only reason they included me in the picture was I was just ending my term as Secretary-Treasurer of ASPET, and I had just gotten through the subprime mortgage drop. And I was so happy not to be treasurer at this point. But it was wonderful, and I think that illustrates how Palmer has fostered faculty and really created a wonderful department over the years. And, you know, it's just kudos to you. It's wonderful. So, congratulations on a wonderful life in science, Palmer, and more to come. So the talk I'm going to talk about, my laboratory for a number of years now has focused on neurotransmitter reuptake systems. We study the transporters using molecular, cellular, and pharmacological techniques to really understand how they work in the central nervous system. not the least of which is the idea that these are targets for major classes of drugs in the central nervous system, including cocaine, amphetamines, antidepressant medications. And my lab has really focused on two distinct families of the transporters. And the story I'll tell you today will link them both together in the end. The story I'll tell you is a fairly small story, and it was largely done by an MD PhD who actually just worked nights in the lab, and Suzanne Underhill, a postdoc, another postdoc, Conrad Hong, and more recently a postbac at NIMH, Patrick Houlihan. And in addition, our collaborators at Oregon Health Sciences and a number of PIC collaborators, including my husband, Jeff Murdoch, who many of you will remember was also a graduate of this program, and I met him in Jeff Rosenfeld's lab during my thesis work. So I just wanted to give you the actions of amphetamine as we sort of often think of it, and certainly most textbooks still illustrate them this way. So amphetamines are substrate for the dopamine transporter, which is the plasma membrane transporter that clears dopamine following its release. And they inhibit competitively the reuptake of dopamine. So they're substrates and they inhibit uptake. It's been proposed that they enter the cell and have additional effects, and those additional effects have been largely attributed to their actions on vesicular transporters, which essentially they enter the vesicle, act as a weak base, and cause depletion of the dopamine and allow it to leak out to the cytoplasm and therefore are depleters of vesicles. And then finally, they elevate cytoplasmic dopamine, and through a series of mysterious events, they've been proposed to cause reversal of the dopamine transporter and efflux of dopamine out from the cell, and that's how they potentiate the actions of dopamine. So there have been a few modifications to these ideas, and I'll start with them. One is that efflux appears not to be simply the reversal of the transporter, but actually has a much more fundamental mechanism that's linked to regulatory pathways within the cell. So efflux requires N-terminal phosphorylation of the dopamine transporter. Efflux requires it appears that CAM kinase binding to a C-terminal domain on the transporter is also required to generate efflux. And more recently, with the availability of the structure of a Drosophila dopamine transporter, we've been able to think about how that might occur. And after seeing all the wonderful structural evidence for mechanisms in terms of membrane proteins yesterday, it's now really thought that the N and C termini of the transporter come together in a fashion, and that the modulation of those domains allows them to come apart and create an efflux pore through the transporter, and that this can be regulated both by CAM kinase, by phosphorylation events at the N-terminal part, and we've recently shown that G-beta gamma binding to a part on the C-terminal part of the transporter, again, allows the transporter to move into an efflux mode of carrier function. So that's simply the background for what we know now. But we became really curious in how all these events would occur in terms of intracellular signaling. And we focused on one particular event, which is known to occur in the presence of substrates for the dopamine transporter. And that is that substrates of the dopamine transporter appear to have the capacity to trigger internalization of the carrier from the cell surface, which is again another indicator that they're acting in other ways to regulate events, signaling events within the cell. And so my laboratory sort of started at this point to really explore what might be those pathways that were involved in this internalization mechanism. And this simply, the next slide simply illustrates the phenomena, which is that amphetamine rapidly induces the internalization of the dopamine transporter from the cell surface. And there's a movie here which you'll see a flash come on, and that will trigger, that's the addition of amphetamine, and then you'll see the changes in internalization. This is using turf microscopy and fluorescently tagged dopamine transporter. And what you'll see is when amphetamine is added, there's a baseline of internalization events, but as soon as amphetamine is added, there is quite clearly an increase in the internalization. And this can be really easily quantitated and by just measuring the number of puncta. And you can see the baseline, there's a low level, and there's a really quite dramatic increase. Removal of the dopamine transporter from the surface, and this can also be measured through activity. We first began to explore whether this was clathrin mediated, because it's the obvious mechanism for internalization. And the next slide simply shows that amphetamine-induced data internalization is clathrin independent. And that is that if you look at fluorescently labeled clathrin and the dopamine transporter, after 30 minutes of amphetamine, you just see no co-localization with clathrin. And this occurs at all time points after. We just don't see the presence of the transporter in clathrin-coated vesicles. And this simply shows that transferrin, which is internalized routinely by a clathrin-dependent mechanism, co-localizes quite nicely with clathrin. So we explored a number of other pathways, including caveolin, which Paul certainly has studied, with a number of other proteins, but we ended up focusing on another kind of mechanism for internalization, which is clathrin and caveolin independent, but it's still dependent on dynamin, which is the small GTPase that's required to pinch off the internalized portion of membrane. And so to explore that, we used botulinum toxin. So here we have toxins that we as well use, and C3, which is a botulinum toxin that inactivates Rho GTPase. Is able to very readily block the internalization of the transporter. And this simply shows the way we did it. This is a dopamine transporter transfected cell next to one which is transfected with both C3 and the dopamine transporter. And if we look, we really see no internalization in the presence of C3. In addition, a dominant negative, Rho A, also prevents this internalization, as illustrated here. And again, C3 very nicely blocks the effect as shown there. In addition, as other Rho-dependent internalization events, inhibitors of cytoskeletal remodeling also block the event. So it has all the hallmarks of being Rho A dependent. Once internalized, this just is looking at EM. We have an external epitope which is biotinylated. The epitope can be biotinylated by an E. coli biotin ligase, which we introduce into the endoplasmic reticulum with the endoplasmic reticulum retention signal. This piece can be readily biotinylated within the ER, goes to the surface, and we can mark it at the surface. So we have a variety of ways of looking at surface DAT and its fate. And in this case, looking at EM, we never see internalization events that are associated with clathrin-coated vesicles, but we do see it in a class of vesicles called tubulovesicular vesicles, which are also linked to Rho A-dependent internalization. The dopamine transporter in the brain has been shown to often be associated when in EM studies with these kinds of tubulovesicular bodies within the brain. In neurons, we can show that these same events occur. Amphetamine causes internalization of the transporter. And this is looking at surface expression in a number of cultured neurons, and amphetamine causes this robust internalization. But one of the interesting features, and I'll highlight this again, is that the internalization is actually blocked by cocaine. So here you have another psychomotor stimulant that also produces hyperactivity and some of the effects of, but yet it's preventing the action. And the way it's doing this is, because we've done a lot of experiments to support this, is that it prevents the entry of amphetamine into the cell. So the actions of amphetamine on trafficking appear to require internalization of the amphetamine via the transporter into the cell, or transport into the cell. And I'll come back to that again. And so cocaine blocks the effect. This is my one slide that's an homage to my bungarotoxin binding days. We also have done experiments where we include a bungarotoxin binding site in an external epitope, and it allows us to essentially label the transporter at the cell surface and watch it traffic through various vesicular compartments. And in doing this, we found that in neurons, that transits through recycling endosomes and returns to the surface. So it's not going to lysosomes, but it really is recycling. So the next question was, does amphetamine activate Rho GTPases in the brain? And to do this, we used an assay for activated Rho. It's a Rho binding domain. Rho binding domain is fused to GST, and in addition, we also looked at RAC1 using a PAC21 binding domain, or a PAC1 P21 binding domain, again fused to GST, and this allows us to identify activated Rho within the brain. And we looked at that in brain slices to see whether amphetamine could activate Rho in a brain slice. And indeed, application of amphetamine within 5 minutes, you see a nice, robust activation of Rho. You also see activation of RAC, as illustrated here, within 5 minutes. And as I mentioned before, these events are blocked by the application of cocaine, which prevents entry of amphetamine into the cell, suggesting that amphetamine's actions to activate Rho require transport of the ligand into the cell. And so we also wanted to make sure that in brain slices that in fact that the Rho was being activated within dopamine neurons themselves. So we used these same probes to layer on a slice to look directly for activated Rho in situ. And if you look at the control there, vehicle controls, in the presence of amphetamine, when we compare the GST to the GST rotekin, which is going to identify activated Rho, the main difference that we see is in the region of the of dopamine cell bodies, which here are illustrated using TH staining in the next slide. So Rho appears to be activated in the dopamine neurons themselves in the brain in the presence of amphetamine. So there's some other interesting aspects that people have kind of ignored about what amphetamine does, and one of the things is that amphetamine increases cyclic AMP production, and this just simply illustrates in SKNSH cells that it does this. But one of the things we noted was that that activation of pKa is well known to essentially inactivate Rho. And so the interplay between activation of cyclic AMP and activation of Rho creates kind of a feedback signal which would both lead to activated Rho and DAT internalization, but also simultaneously would also have an influence to inactivate Rho. And so we began to explore this because in some ways it's interesting to imagine that you could tap into this to actually prevent the effects of amphetamine on the dopamine transporter in terms of its ability to internalize. And so that pathway. So what we did was we used we looked first for phosphorylation of row A and saw a significant increase in phosphorylation at about 30 minutes. This is actually a turf assay using wild-type row, a dominant negative row where you see no increase in, you see no increase in internalization. And if you use a pKa insensitive row that's been mutated in the position of that the phosphorylation site, you can see you actually potentiate internalization, suggesting that indeed cyclic AMP is either is definitely regulating and has the capacity to really shut down the effects of Rho on the DAT internalization. And so, as you would expect, cyclic AMP has altering cyclic AMP has effects on DAT internalization. And so, if you increase cyclic AMP or pKa activity, you essentially block internalization. But if you inhibit pKa, you actually potentiate internalization. Again, fitting into the model that I've just described. So we went on to really look in cell lines to see whether these events could be triggered not only by amphetamine entering the cell and increasing cyclic AMP, but could you get cell surface receptors to actually modulate the effects of amphetamine? And so here we, in SKSH cells, which have a number of different receptors expressed, and we introduced the dopamine transporter and looked at internalization. Amphetamine produces a very robust internalization, but activation of cell surface receptors that couple through G sub S essentially prevents these events. And we could also do this in primary TH-positive neurons, where application of a compound that's a D1, D5 agonist could essentially inhibit the internalization mechanism. And so I'll bring you to just a first summary slide at this point and just illustrate that what I've shown you is that cell surface receptors acting through G sub S have the ability to modulate cyclic AMP and turn off the Rho-dependent internalization mechanism. Amphetamine entering the cell leads to an activated Rho and also increases in cyclic AMP. And the interplay between these pathways really determines the internalization pathway. We also tested this sort of model in a behavioral assay as well. And what you can see here is that, in this case, this is a mouse line that had been administered amphetamine and cocaine. And some were pre-treated with a D1, D5 agonist that would increase cyclic AMP in those cells. And what you can see quite nicely is that, that in the case of amphetamine treatment, SKF 38393 has the capacity to really inhibit acute effects of amphetamine on, in this case, locomotor behavior. But it really had no effect on cocaine, which is another psychomotor stimulant that would be elevating dopamine in similar regions. And so we also showed that, in fact, we could see changes that amphetamine administered to the animals also produced a decrease in RTI binding, so the number of dopamine transporters, but at the surface. But it also could be blocked by activation of cyclic AMP pathways. So it's preliminary behavioral data, but it supports the model. So there are a lot of unresolved questions here. In my last few minutes, I'd like to get to them. The first one is, how does cyclic AMP activate Rho? And then the second one is, how does it increase cyclic AMP? And so we've explored that further. And one candidate for an intracellular target for amphetamines is a trace amine-associated receptor, which is expressed in dopamine neurons called TAR1. This is a receptor in a GPCR, and we had studied it a number of years back when it was originally cloned, and it responds to a number of trace amines, biogenic amine metabolites, ergot alkaloids, a number of drugs. And so it has a wide variety of different agonists. It responds to beta-phenylethylamine, tryptamine, and it also is coupled to increases in cyclic AMP. And this is in surface expressed receptor. And there's also evidence for GQ coupling. It's predominantly intracellular, and that was the most intriguing thing. We actually had trouble getting it out to the surface. And so we thought, because it's present in dopamine neurons and because it is intracellular, we began to explore the role of TAR in the actions of amphetamine on DAT trafficking. And this just simply shows that in a CRISPR-Cas line, if you delete TAR1, this has TAR1 present. If you delete TAR1, you lose the effects of amphetamine and on trafficking. And if you replace it with TAR1, it goes back. In TAR1 knockout animals, what you see is that the genotype correlates quite nicely. You see an effect of amphetamine on cell surface DAT, and in the presence of with the heterozygote, you see a reduced internalization, and in the homozygote, you see very little internalization. And similarly, Rho activation correlates very nicely, suggesting that Rho activation may indeed be linked to the activation of TAR within the cell. And this simply shows the Western blot that was generated from. So I just have a couple more minutes, but I'll come back to what I've told you and summarize a little bit more. So again, what we see is that activation of cell or amphetamine entering the cell likely activates TAR1. We've shown in a number of other studies that it's actually through G-alpha 13, and that leads to the activation of Rho and regulation of cell surface expression of the carrier. In addition, cell surface receptors have the ability to modulate this by regulating cyclic AMP action. And so I have one more short bit to tell you after this to bring in glutamate transporters, but amphetamine acts intracellularly to activate Rho, RAC, and to trigger Rho-A-dependent internalization. It doesn't actually depend on increases in extracellular dopamine. So we see these effects of amphetamine in HEC cells where they don't make dopamine nor do they release it. So this isn't a cell surface receptor phenomena through the potentiation of dopamine. It's really something that happens within the cell. In dopamine neurons, PKA activation via endogenous GS-coupled receptors can inactivate Rho and inhibit the acute behavioral effects of amphetamine. And intracellular TAR1 appears to be a link in this whole pathway. So we went on to look at one more aspect of it, and hopefully I'll get through this in the next minute or two. But we also looked at a glutamate transporter. So many of you know that dopamine neurons are much more complex than we previously thought. They're more heterogeneous, but they also are glutamatergic. They release glutamate in vesicles. They have transporters, both vesicular and plasma membrane for glutamate. And so we asked whether or not the plasma membrane carrier for glutamate was regulated in a similar fashion. And indeed, what we observed was that the endocytosis of a glutamate transporter, EAT3, expressed on dopamine neurons, was internalized in response to amphetamine, and that internalization is dynamin-dependent and Rho-dependent as well. We went on to look at what the consequences were for excitatory signaling. And amphetamine increases the amplitude of stimulus-evoked currents on dopamine neurons. So this is activating terminals that release glutamate into the VTA and reporting from the VTA in dopamine neurons. And what you can see is that in the presence of amphetamine, you see a potentiation of NMDA and AMPA currents. And you see it kind of, it's agnostic to the receptor type, which is kind of consistent with it being potentiation that you would see by blocking reuptake through internalization. So we identified an amino acid sequence on the C-terminus of the EAT3 transporter that essentially was required for this internalization, but not for the internalization of DAT. This allowed us to make a TAT fusion peptide that we could introduce into the cell to block the excitatory effects of amphetamine. And so in this case, we could show that a TAT peptide fused to this C-terminal motif essentially could block the potentiation of glutamatergic currents, but not, and it didn't have any effects on the dopamine transporter, this particular peptide. So I'll just go on to say one more, amphetamine doesn't potentiate NMDA EPSCs in EAT3 knockout mice, suggesting that EAT3 plays an important role in the effects of amphetamine. And we can also show that the acute behavioral effects of amphetamine are markedly disrupted in EAT3 knockout mice. And so I was going to say one thing about meth, but I'll end on my summary, which is what I've tried to show you is that the effects of amphetamine presynaptically on dopamine neurons involve the uptake of amphetamine, and they regulate both endocytosis of the dopamine transporter and a glutamate transporter, thereby modulating both dopaminergic and glutamatergic signaling. The actions of amphetamine lead to adaptations that may determine the unique effects of amphetamine compared to other psychostimulants. And I'd like to say that, in terms of amphetamines, we give about, there are about 3.5 million children across the country who take medications for ADHD, and about more than half of them are on Adderall. And so it seems like we should understand what these drugs do within the cell. I didn't tell you that meth can enter the cells in the absence of DAT, and we've seen that it has the same effects on glutamatergic transmission, but more broadly. Therefore, it acts more broadly than amphetamine. And finally, that these signaling pathways are likely to be the same ones that respond to endogenous amine transmitters and those within the cell. And so these pathways actually can also be activated by dopamine, by various metabolites and other ligands that really interact with the TAR receptor. Finally, I wanted to acknowledge the rest of my lab. This is my lab at NIH, and thank you all for your attention. And again, thank you, Palmer, for the inspiration that you've given so many of us for the past many years. So thanks.
Thank you so much, Susan. I think we have time for maybe one question, if there is a burning such question in the audience. Anyone? Yes. He's going to bring you a microphone, just a second.
Is there anything known about what regulates the density of transporters at the synapse itself?
So I should say the transporters are really not in the synapse. They're actually, most of them are perisynaptic and localized, and they're really poised to regulate spillover much more. These mechanisms for regulating trafficking would occur, again, in these domains that are probably extrasynaptic. So.
Thank you so much, Susan. And it's my pleasure to introduce the next speaker, Craig Venter, who essentially needs no introduction, I would say. to this audience, an old friend of UCSD, having been an undergrad and one of the first graduate students in what was then the physiology pharmacology program. And Craig's going to talk about human genomics and the future of medicine. Craig?
Palmer, it's a pleasure to be here and celebrate your half century or more of work. I met Palmer and Susan in 1971 when they basically had just arrived here. I think thanks to Palmer, I became a graduate student here. I think I'm the first and last who snuck in the back door with Palmer's help and Nate Kaplan's help. So, but I left here a little over 40 years ago and started to come back 10 years ago. So it's been a long journey over the last 4 decades, but they've been exciting ones, and I'm going to talk to you about how they're all coming together right now. So thank you very much. Okay. All right. So I'm going to talk to you a little bit about human longevity, but first let me mention sort of what. That's okay. The keyboard works just fine. Hopefully you've seen this building. It's less than a half mile from here. And it's sort of what helped bring me back here starting a decade ago. We opened this building now a little over 3 years ago. But the work at the Venture Institute, which is a not-for-profit, has led in a number of different directions. But with a common goal for a very long time, and that's trying to predict phenotype from the genotype, and trying to work out whether we can in fact predict all future programming of species, including humans, straight from the genetic code. It's interesting, this is a study we published now a little over 20 years ago. This last summer I talked to about 50,000 high school students interested in science and medicine, none of who were born or existed when this paper was published. So it starts to get humbling when you become part of history without realizing how many new generations there are in science. But even more surprising, so 15 years ago was when we published the first version of the human genome, and I would say at least a quarter or more of the scientific community was not in science 15 years ago. So it's a rapidly changing dynamic, which I think constantly leads to new ideas. So even though I started work here at the single neurotransmitter receptor level, we're now trying to integrate things across the entire human species. And 4 trends sort of converge to bring us back and lead to the start of human longevity. It's one, the reduced cost of sequencing, so that genome that we published 15 years ago cost $100 million and took 9 months with a substantial group of people. It's now down below $2,000 for a genome, so that was a key change. Computing, 15 years ago, we spent $50 million building the 3rd largest computer in the world, and it was only 1.5 teraflops. So you can, for $100, buy a card for your PC now that does the same. And with distributed cloud computing now, we basically have close to infinite computes, although recently we've been using Amazon Cloud, and I mentioned that at a public meeting where it was being filmed that we were using Amazon Cloud, and we were in the 1% group, so HLI's up there with video streamers and porn sites now. But we're the only ones using a lot of CPUs, and we needed 10,000 extra processors to redo a calculation on 10,000 genomes, and Amazon couldn't provide them on a stat basis. So I said we blew a hole in the Amazon cloud. and they cut it off my video at that point, but they used the rest for an advertisement. So, but cloud computing is key, but computers are still somewhat limiting in this space. And the third component that has been really key to what we're doing is new types of computing. So machine learning is one of the most interesting areas. I hired Franz Ock out of Google. Many of you know what he built. He built Google Translate. He did it all over 100 languages into each other with a small team using machine learning. So I convinced him that the human genome was a bigger translation problem. So about 20 months ago, we started Human Longevity. We now have 260 employees over in the UTC. And we also have a site in Singapore, and the machine learning group is in Mountain View. And the goal is to essentially bring genomics together with all the phenotype and clinical data that we can to use machine learning to make multiple predictions. I'll show you in a minute. We're trying to predict faces right from the genetic code. We can predict age now. We're trying to predict disease risk, life expectancy, et cetera. We've been working with Palmer and the School of Pharmacy here. We've had a number of interns that have been helping us annotate the genome for pharmacogenomic effects. And Palmer, I don't know if they ever told you this, I asked them the number one thing they learned at the end of their internship. They said they learned that the scientific literature was absolutely worthless because it's so variable and so full of noise that you can't get any reasonable facts out of it. So instead of using the literature to build up the annotations of the genome, we're using our database actually to validate the few things that are right in the scientific literature. So we built a large sequencing facility. Just since inception, we've now sequenced over 20,000 human genomes at high coverage. We're doing about 3,000 a month, but scaling up in a lot of spaces. So we've just recently submitted a paper on analyzing 10,000 full genomes to see what the accuracy of the technology was, what we could get out of it, et cetera. And it showed a number of things. When people talk about sequencing the human genome, we're still only covering about 85% of it in any kind of accurate fashion. It's because of all the repetitive DNA around centromeres and part telomeres. The other thing that we found is around 8,000 human genomes, we've saturated all the common variants in the human population. And I'd say that was one of the biggest surprises to us. And so now if we sequence your genome, we'll find about 8,000 new rare variants. We have now over 80 million rare variants that only occur in our database one time. And these are going to be the ones that we think are most important for understanding our own physiology. But even the scientific literature is now confusing on this. 15 years ago or 20 years ago, it was very clear-cut what a genome was. You had to have every base pair sequenced, no gaps remaining. And then with the human genome, it started to slide. Even when we assembled the human genome the first time, there were still gaps around the centromeres. But recently, a paper was published in Nature saying it was describing 1,000 genomes. It turns out those genomes were only sequenced to 7x coverage, but it's really horrible nomenclature because 7x, when it's done with Illumina with very short reads, only covers about a third of the genome. 7x coverage with earlier applied biosystems or long read technology gives you high coverage of the genome, so you have to know the context. So it sounds impressive to people until you see where this line is on the left. That was what was described in the Nature paper. So it was sequence data from 1,000 genomes. It wasn't actually 1,000 genomes. And with the 10,000 genomes, our goal is to have coverage high enough where we cover every base pair that's interpretable at least 10 times to get accurate data. So a map of the genome now looks like this. I don't know if those little tiny blue marks are vertical measurements, and you can see sort of the gray areas around the centromeres where there's a low coverage. But this data is important to the FDA because you can see down at the bottom, the false positive and the false negative rates are extremely low, which means that we have a chance of making accurate interpretations because we're starting with accurate data to start with. We're also sequencing exomes because in addition to the higher cost efforts, we have 4.4 million clients now in South Africa and in England that wanted to start a lower price point of $250. So you can see the scarcity of the blue lines there. The exome covers about 2% of the genome. That's primarily just the coding regions of genes. And right now that covers about 85% of medically known actionable data in the genome. But to put things in context for people who get confused with 23 and others, so gene tips measure what's in that little box on the far right. Those are the common variants in the genome. And as I said, we're just adding more and more extremely rare variants. And we have a wonderful search engine that we'll be bringing out in the public later this year that if your genome is sequenced and you type in your name, it'll bring up all 3 to 4 million variants in your genome in about 30 milliseconds. And you can search any gene for its frequency in the entire database in 10 to 20 milliseconds. So the computes and the changes of what's possible are affecting things quite a bit. So the goal is to take data from the genome, from all the phenotype data, from health records. And in particular, we're trying to use machine learning to turn that into knowledge. And the goal of Franz's team is to predict anything that can be predicted. And he's building a machine learning engine where we can ask any question of the data. So the limitations going forward are coming up with the right questions, because no matter what the discipline you come from, it brings up lots of unique ones. To show the power of the machine learning and what's contained in the genome, I asked him to start to see if he could predict age from the genetic code and also faces and portraits. Age is an unusual phenomenon in the sense that if you'd asked me 15 years ago, I'd say you get your genome sequenced once and that's it unless you have cancer. And it came up to the huge surprise that we can pretty accurately predict how old you are, or at least how old you were when the sample was drawn. And it takes a number of parameters into account. For example, as we age, we lose X and or Y chromosomes. You can see there's a lot of scatter in these plots, but in conjunction with other data, it becomes very clear-cut. So maybe that's why men get nicer as they age. They're losing their Y chromosome. I don't know. But I think a lot of people are surprised by this. And then the other thing, the accuracy of our sequencing with a new algorithm, we get very accurate determinations of telomere length, more accurate than by any other measurement. And it does correlate nicely with age, and that helps quite a bit. And then there's features in the mitochondria, the loss of copy number variants, losing segments in your mitochondria, actually losing mitochondria. There's a lot of different features that change dynamically with age. So we can put these and copy number variants across the genome together to get a prediction. I mentioned FACE, so we actually did a clinical trial starting mainly here at UCSD where we recruited students. It was actually, we thought we were recruiting students. We made the mistake of starting to pay $100 a student for a sample, and it spread very rapidly to their families and communities. And we had a problem because we had a high percentage of twinning. And it turns out some of these people would go change their clothes, come back and claim they were the twin of somebody. And we got pretty good at catching them, but a couple of them came through and we didn't catch it till we compared sequences. And then it turns out they're sort of chronic clinical trial people. So their same genome showed up in multiple clinical trials here at UCSD. So you start to learn a lot about people with this kind of information, but now we insist that twins come in together. So it's a little bit more difficult for them to fake it. We wanted to see if we could predict the voice and your face from the genetic code. We started with a number of predictions from voice, and we're quite surprised. So now from a simple recording of your voice, we can readily predict your age, your sex, and surprisingly your height. Height is probably just a surrogate for your volume that relates to voice. But you can see it's pretty clear cut. There's, like everything else in the world, there's sort of this ambiguous zone on sex, so there's nothing surprising with the voice either. But these now are starting to correlate with structural features in the face as well. So we're still working on predicting the voice straight from the genetic code. We've been much more successful with the face. So it turns out our predictions predict how you look just post-puberty. So trying to find younger pictures of people without beards was difficult. So here's the straight computer prediction, and just trying to line it up, it helps a little bit. But here's a much more complex situation. Ethnicity predictions are a key part of things, but you can see this is a woman with complex ethnicity, and the actual photographs are on top and the predicted is on the bottom. We take very high density 3D photographs, and they're so intense, they're not necessarily face friendly. So you can see the original photograph on the left, we smooth the data, and then you can see the predicted. People like their predicted pictures because right now the algorithm makes people look totally symmetrical, plus we smooth out all the other parts. So, and as you can see, the predicted does look younger than the current photo. We now have a nice algorithm that, based on predicting your age from the genome, will appropriately age the photographs. And this is an early version, the latest version that just came out last week. They're starting to look much more like actual photographs. So here's another one. So you can see we're starting to make progress. These pictures don't include the eye color. We can predict your eye color extremely accurately, in fact, more accurately than you can describe it, and also skin tone. So we're collecting data in a variety of ways, and I'll go through some of these. We do whole genome sequencing. We have a very detailed, comprehensive cancer program, and everything from sequencing newborns to pediatrics with rare diseases. So I mentioned that we have this deal in South Africa with Discovery Health, and they're adding exome sequencing to every one of their clients, and they've put together a novel report trying to predict heart disease, cancer risk, and a variety of other inborn errors. We started just as something just to help a friend, and it turned into this comprehensive cancer analysis program where we sequence the whole genome on the patient, the whole genome to very high coverage on the tumor. We sequence the RNA from the tumor. We do immune-seq, which gives us the T-cell repertoire. We can do this either from the blood, but we prefer to isolate the T-cells from the tumor. We also do liquid biopsies of measuring circulating DNA as well as circulating tumor cells. And this expanded to now cover a lot of different cancers in collaboration with a lot of centers, most particularly here with the Moores Cancer Center. But what you get when you have the genome of the germline as well as the tumor, you can immediately calculate the mutation burden. This is an HPV-16 caused throat cancer, which is somewhat of a pandemic primarily in men in their early 50s. In this case, we found 25,000 mutations across the genome. 315 of those change protein structures, and we can verify these with RNA. With these HPV-16 tumors, we find the virus integrated in the genome in multiple sites. In this case, it was 4 sites. We just did one where it was 16 sites across the genome with the virus integrated. And it's not known whether the HPV vaccine would work to help prevent these cancers, but it's certainly worth trying if you have high levels of HPV-16. We also do a comprehensive pathway analysis, so we take the mutated genes into pathways to find other druggable targets in these same pathways, and it's coming up with a lot more information than the other existing cancer panels that are out there. In one particular case study, there were 45,000 mutations. We came up with 8 novel drugs for clinical trials, and there were 39 neoantigens, so the high mutation rate. But what we did is we made peptides from those, selected the T-cell repertoire, used those for expansion with immune therapy, and they've been actually quite effective in this one individual that's going extremely well so far. So you can see with different types of tumors, we're seeing different mutational burdens, but we're hoping to see patterns across the board as this develops. But the main program that we're trying on a therapy side, other than predicting novel drug combinations, is to take these in to develop both the T-cell kind of approaches, but just making RNA and peptide vaccines. And our goal is to go from getting the tumor sample to 3 weeks later having a RNA vaccine to give to somebody in conjunction with other therapies. So we're starting this, and we're doing this in collaboration with Ezra Cohen here at the Moores Cancer Center. So getting accurate phenotype on people was really an issue, so we formed what we call the Health Nucleus to create a unique phenotyping center where we bring all kinds of information together, but we do a lot of our own direct measurements. So here's a partial list of all the things that we measure in an 8-hour visit. So it's an intensive day for people. We sequence the whole genome, we sequence the metabolome, we do GI microbiome. In terms of chemistry, we measure over 2,000 different substrates in the blood, and then combine that with all this other direct imaging data. This is our new 3T MRI machine that has protocols that only we in GE Research have, and some of these new protocols are absolutely stunning. So this is the brain vasculature on one of the people who've gone through. It might be my brain, I can't tell you for sure. And this is without contrast. This is just the new water differences in MRI, just the aqueous difference in the blood vessels versus surrounding tissues. The same things work for detecting tumors. So this is about a 10-minute scan in our MRI with no contrast media, nothing. Unless you get claustrophobia, you may need some benzodiazepines to stay in there for a while. But all our scans right now are taking well over an hour to go through all the processes, but using some really phenomenal tools that were developed by Anders Dale and his team here at UCSD called NeuroQuant, where they get quantitative volumes of different brain regions. It's very easy to find some of these areas, and it's quite easy to pick out the hippocampus here, if you guys didn't already know where it is. And so you can see with clear-cut Alzheimer's disease, the clear loss of hippocampus. It's not just loss of the volume, you can see these spaces around it, and these become very easily detectable. It's not only detecting the disease that's important, it's detecting the absence of it. So this is a 69-year-old female with a family history of Alzheimer's disease and obviously very concerned about it, who has a very large and robust hippocampus that I'm sure you can see. In fact, people get these quantitative reports. She was actually in the 99th percentile. Every organization has its own things that they measure. We have two. We compare people based on their hippocampal volume and their liver fat content. So Franz Ock right now has the largest hippocampus of anybody, which might be predictable given his unique approaches. But what I like to consider, people that have the really large hippocampus have to give up some other part of their brain for that same volume. So, you can't have it all. So we get these very quantitative metrics, and the other thing for the computes, it takes a couple gig file and converts into a simple table of brain volumes that makes it easy to take into other approaches. So this is this restricted spectrum imaging that teams here in oncology have really developed, and it's absolutely stunning. So again, with no contrast, they can now accurately diagnose a prostate cancer without anything else, and it correlates very nicely with pathology when there was surgery. So problem with doing MRI imaging and CT imaging in the past was all the incidental omas that were found. Now with this new restricted spectrum imaging, we can clearly discern tumors from non-tumors. And you will see that we're finding things in 30 to 40% of people that come in that think they're perfectly normal. But you can see these things light up very clearly. Here's a angioma. I don't know if you can see it right on the top of the brain. It's a walnut-size piece. That most of these are benign, but they also have a bad tendency to bleed with any kind of head injury. So this individual is having it surgically removed. To show you the type of imaging, so this is 56-year-old male who has been totally normal, except he sort of had this rare hoarseness, and he would go and get annual physicals and chest x-rays each year. The chest x-rays probably made this worse, but you can see the tumor just lit up like a light bulb with this restricted imaging. The radiologist diagnosed it as a thymoma. The surgeons used our MRI images to robotically remove this tumor. It hadn't penetrated the tissue, so basically he went home the next day cancer-free. And the pathology confirmed that in fact was a thymoma. Had it progressed to stage 2 or stage 3, where it would have created symptoms for detection, it would have, he'd probably have a year left to live. You can get exact quantitation of your internal fat versus your peripheral fat, your exact volume of different muscles, so you can measure that health. As I mentioned, we can detect liver fat very quantitatively. This is somebody with a very high level on the order of 26%. 5 is normal. Mine went from 4 to 7 over the holidays. The important thing about liver fat, it does change dynamically if you change your behavior dynamically in either direction. This is an ovarian cyst that was detected in another normal individual that had no awareness of it whatsoever. And even if these aren't cancerous, they can actually twist. And so torsion with these can lead to death from bleeding. From CT imaging, this is actually my heart. So you can see the image qualities are stunning from what's coming out these days. And we can get exact quantitation of amount of blockage in coronary vessels. And then we use the new 4D echo ultrasound. And I think from this movie you can see all 4 chambers of the heart here. it'll turn and look right down the valve. We can measure Doppler shifts, so the physicians at our clinic are not allowed to have stethoscopes because we can detect the murmurs that no physician can hear with a stethoscope just looking at the Doppler shift in the blood movement. So, here's the news that's affecting all of us. If you're a male between 50 and 74, you have a 30% chance of dying in that time period. If you're a female, you have a 20% chance of dying. Roughly a third in each case is from cancer, slightly higher with women, and roughly a third from cardiovascular, slightly lower with women. So while we knew about these statistics, when we actually started to find this in normal, healthy people, it became surprising. So here's just some of the findings early on. Again, most of these are executives or physicians. Some of the findings in them. The good news is all of these are actionable, and like that one tumor, treatable. So you can see we're finding a fair amount of NASH. With the 4D echo, we found 2 dilated aortas, one in an individual who was uncontrolled hypertension and a heavy weightlifter. So he was just waiting to have a blowout, so he said surgery to correct this. But the exciting phase is when we put all this together, we're finding phenomenal genotype-phenotype correlations, including new ones that nobody ever knew about using the machine learning. So we found a nice gene duplication event in the genes that we can now start to predict aortic dilation. Obviously, we're generating a lot of data on each individual, and the challenge is reducing it. I mentioned how we reduce the brain MRI images. We take full body 3D photos and we create avatars, and you'll get all this information back in an iPad where your avatar will walk you through all your information from your skin all the way through to your skeleton and to your genome. So we're trying to integrate all this data across as many as 1 million samples over the next few years and see if we can actually change the predictive nature and the preventative nature of medicine. Thank you very much.
Thanks, Craig, for a terrific look to the future, look to the present and future. Time for a question. Jean-Pierre?
Thank you very much for this beautiful presentation. Actually, there are a lot of data which are present in the non-coding sequences of the human genomes. And one of the challenges is to try to identify regulatory sequences within these non-coding sequences. And in fact, when you look at the evolution of the coding genome from mice to humans, there is very little change. So we have to find the change elsewhere. So my question is, how far are we in the identification of the regulatory elements present in the non-coding sequences?
Yeah. Thank you, Jean-Pierre, for the question. Most of the new findings we're making are in the non-coding part of the genome. There's a paper pending that we submitted on these 10,000 genomes that in fact show areas of the genome that cannot tolerate any mutations and be compatible with life. So we've identified those. A good number of them are actually splice sites for intron-exon boundaries, where fundamentally we just don't see any variation at all on those. But we're trying to show, if we sequence your genome and give you a report, you would get a report back of what percentage of your genome is in microRNAs, introns, etc. And the regulatory part of the genome is, I think, where most of the new discoveries will take place. So people focus on the protein coding regions out of bias and because they could. And now that we have the whole genome or the key parts of it in a very accurate form, we can now make nice predictions in these areas. So I think it's going to be very exciting over the next few years.
We'll take one more question way in the back on the left. I can't see you.
Is there any plan to make this data all available? So I think that was the power of the human genome, and different people are going to look at this data in very different ways. And is there any way to make it accessible like the genome? Talking about the imaging and the whole cohort of data.
So we are putting together a database that will be available in a variety of fashions, but now that we can predict your face from the genome and other physical parameters, we have an obligation to keep people's identity highly confidential. In fact, with simple triangulation, you can usually identify somebody. There was a paper published last year that just used my Y chromosome haplotype information to link that to my surname to finding out information about me and my relatives. So we want to make it certainly accessible and to do searches on it. It's very easy to make links and find identifications of people. So we're trying to do it in a way that will guarantee people's anonymity going forward.
Thanks so much, Craig. Our next speaker before the refreshment break is Joan Heller-Brown, who's a distinguished professor and chair of pharmacology here at UCSD and has been at UCSD for a long time, especially with Palmer. And her topic of her talk is novel targets for GPCR and ROA-mediated pathophysiological responses. John.
Great. Well, it's really exhilarating to be here and to see all of the people who Palmer has trained come back and reflect the training that they got here from Palmer and also from the graduate program. To see the breadth of their knowledge and what they've achieved and where they've gone is just a wonderful tribute to Palmer. And I want to start by also giving a tribute to Palmer, and then I'll get into talking about what I think I can actually say about toxins to therapeutics. So Palmer means many things to me. I came in 1975, so I've been here for 40 plus years. Palmer was my division and then department chair for a number of years. He's been a collaborator as well. We've actually published some papers together and published a lot of chapters together at the time when I worked on acetylcholine, and we published papers in several editions of Journal of Neurochemistry and Goodman and Gilman on that topic. Palmer also, this is supposed to move forward, no? Can you, yeah, that's supposed to do it?
Yes, you can use freezing up on you. There you go. So which one did you like? This one?
Go back. That's the next one. No, go back.
There you go.
Yeah, that's the next one, but why, it just froze?
Yeah,
Okay.
right, so it was up on there.
Okay, so Palmer has also been a teacher, as he has been to many of the department. I think since I got my PhD and did my postdoctoral work in pharmacology departments, probably Palmer assumed that I knew lots of pharmacology. I knew a little bit of pharmacology, but attending his lectures and learning what he knew really was an incredible and useful experience. And he has been what I'm referring to as my patron saint in many ways. He supported me all along my career, having me be his vice chair and interim chair, and then taking over the training grant, and created a department that I was able to build from in my current role as chair. In the spirit of this symposium, Toxins to Therapeutics, I actually realized that my talk relates to toxins in a very evident way, and that is that very early on, in almost 30 years ago, I had a postdoc in the lab. Somebody here might remember him, Palmer might remember him, named Lawrence Quilliam. He's kind of a difficult guy, and he was working on a really odd problem. He was convinced that G proteins, small G proteins, regulated phospholipase, and had some studies that said the toxins regulated, botulinum toxin regulated phospholipase. That was no way that those things were related by anybody else. And we did a study, actually, he published some of the stuff on the phospholipase regulation, but we did a study with Jan Buss, who many of you know was also a graduate student here. And Jan actually, we tried to see what it is that was being ribosylated by this toxin from Clostridium botulinum. And Jan isolated a 22 kilodalton RAS-related protein. At the time, there was no notion of what that was. RAS was known as a small G protein, but its other family members were not. And then subsequently, Lawrence, working with Gary Bokash, actually showed that that protein was what we now know as the small G protein. So we actually worked on this small G protein before it was known, and these studies were carried out in a cell line that we've studied for a number of years. I will just say that this toxin now is well known, well studied by Actories and others, and it is the C3 exoenzyme from Clostridium botulinum that ribosylates and functionally inactivates Rho. So in our studies on these cells, kind of fast forward a few years, these cells that had this, turns out, this ribosylated Rho protein, we were looking at cell proliferation. And we had been studying the muscarinic receptors in these cells. They couple to GQ and regulate phospholipase C, and that was sort of my focus at the time. And Silvio Gutkin, who I think is not here but is now on our faculty, published a paper where he said muscarinic receptors are oncogenes. And I thought, Jed, that's really cool. I'm going to see if the muscarinic receptors actually stimulate proliferation in these cells, which happen to be a human line of astroglioma cells. And we looked at the effect of carbachol acting on the muscarinic receptor. It stimulated BRDU and thymidine incorporation a little bit, but really not very well. What did an amazing job was thrombin. I actually can't remember why we were working on thrombin, but Joanne Trejo was in my department, in my lab as a graduate student, and she was studying thrombin actions. And so we went on to try to understand what it was that thrombin was doing. And what we discovered was that thrombin, unlike carbachol, was a very good activator of Rho-A, and Susan introduced the assay for active Rho-A. And furthermore, that if you blocked Rho-A signaling with this very useful tool, you blocked the proliferative response. So the proliferative response was regulated through Rho-A signaling. We knew it wasn't regulated through GQ because carbachol didn't do it. We knew it wasn't regulated through GI because pertussis toxin didn't block it. And so we kind of thought, how is it possible that this thrombin, we now know, and we're learning about that time, that it acts through a G protein coupled PAR1 receptor. How is it that thrombin is able to activate Rho? And then later I'll talk about how Rho regulates proliferation. But at around the same time, and many of you are familiar then with the G protein family, Mel Simon's lab had found the newest member of the heterotrimeric G protein family, which are G12 and G13. And their functions were really not known. But Mel and also Silvio and also Aronson at the same time had all shown that this G12 protein was actually initially isolated as an oncogene and could serve as an oncogene. So we Mel was at Caltech at the time. Mel was also down here for a number of years. Many of you know, lab next to mine for a bit. But Mel was at Caltech at the time, and I talked to him and said, this would be really interesting to see if this G12 protein is what's allowing thrombin to talk to proliferation. And Mel sent down Anna Arragay, one of his postdocs, who came to our lab and did this experiment showing that G12 blocks thrombin-induced proliferation. You'll note that how this is done is with an antibody that they had to G12. And this was back in the days when we didn't know how to get anything into cells by transfection or virus. And it was done using microinjection, which Jim Fermisco, a member of our department as well, a technique that he pioneered. Linking this all together was really made possible by studies that Paul Sternweiss's lab did, and they actually showed that the effector for G12 and 13 here was P115, but we now know there's other ROGEFs or ROGEFs, whatever you like to call them, that actually bind to G12, 13, and in the same way that GS binds to and regulates cyclase, GQ binds to and regulates PLC, G12 and 13 bind to and regulate ROGEFs and activate Rho. So that's how GPCRs talk to Rho. And there's only, if you try to look in databases and figure out which ones do talk to G12 and RhoA, you won't get a whole lot of information because it's not well annotated, but for certain, the Part1 thrombin receptor, the receptor for sphingosine-1 phosphate, and I'll talk about both of these sort of interchangeably, the receptor for LPA, so these lysophospholipid receptors, are very efficacious at coupling to G12, activating Rho, and stimulating then Rho-mediated responses. And so knowing all this, we've spent probably the last 2 decades in various ways trying to ask, what is it that's downstream of Rho? How does Rho signal to elicit proliferative and other, what I now refer to as sort of pathophysiological responses? In other words, how do GPCRs that activate Rho do things within the cell? And I'll tell you mostly about a story about the proliferative response here, and we have data on a lot of other of them, but just to sort of tell one story on this, actually what Joanne, one of the things that Joanne showed in the lab was that if you stimulate the cells, these same cells with thrombin, or carbachol, the thrombin is much better, it's actually much better at increasing c-Jun, which is a component of the transcription factor AP1. And this shows that thrombin is very good at activating AP1 luciferase. We learned this from Michael Caron in the department, who was actually on Joanne's committee. And this happened to be the gene readout that we assessed. But the bottom line was that, thrombin, unlike carbachol, is very good at activating, at turning on genes. And so we went on from there to say, well, what genes does thrombin turn on that carbachol doesn't? And we ran some microarrays. And of course, we found many, many genes, and we did a variety of times, and we always compared thrombin and carbachol, and we came out with one that it took us a long time to appreciate, and most people still don't, but it was a funny gene. Everybody finds it in their arrays, actually. It's called CYR61 or CCN1. And this shows basically that it goes up very rapidly, that it's still up at 12 hours. This is the message level, but we've done protein as well, of course, and that it really doesn't go up very much with carbachol at all. So we set out to learn more about what this protein was, and it turns out in some other people's literature, there's a ton of literature about CCN1 in cancer for certain. But actually, before we did that, we also showed that this response that happened with thrombin but not carbachol was mediated through RhoA signaling, again blocked by our C3 toxin. So CCN1 is referred to, it's in a family. There's a CCN2 and a CCN3. CCN2 is CTGF. Somebody might have heard of that. I'm sorry, these backgrounds, with the light, it's really hard to see this. Maybe better on this side. So CCN1, but I'm supposed to point to this one because they're videoing.
No, it's okay? Okay. So CCN1 is referred to as a matricellular protein. It's actually secreted from the cell. It doesn't have a receptor of its own. It binds to heparin sulfate proteoglycans, and it interacts with and activates integrins. And so we asked whether this pathway from the thrombin receptor to Rho and CCN1 gene expression was involved in the proliferative response, and indeed it is. And here we've blocked, using an antibody to CCN1 outside of the cell, blocked the thrombin-induced proliferative response. And then I told you that it's also acting through integrins, and we did a similar sort of thing using an integrin antibody blocking the integrins that we had determined were important on the surface of these glioblastoma cells, beta-1, alpha-5 beta-1, and showed that the beta-1 integrin also blocked the cell proliferation. Another way that integrins are activated, and I'm kind of trying to tell you that GPCRs tend to act through integrins. There's another way that integrins are now known to be activated. Sandy Chattil, Mark Ginsberg, among others, have worked a lot on this, and that is by something called inside-out integrin activation, and RAP through binding to some other proteins that are not shown for simplicity's sake here. RAP actually activates integrins from the inside out, and we showed, and I'm not showing you, that thrombin activates RAP in a potent and sustained and way, and it does it through Rho. And we asked whether RAP was actually involved in integrin activation as well in response to stimulation with GPCRs. And we looked at this by looking at, sort of slightly indirect, but looking at integrin activation by looking at the ability of the cells to stick to fibronectin, adhesion to fibronectin, which is integrin dependent. So here's the control cells, and here's the cells when you treat them with thrombin, and you wash them in either case, and the thrombin cells stick much better to fibronectin. You know, that's shown here, or shown here, is what thrombin is doing in terms of measuring the absorbance. But if you knock down RAP with SI or shRNA, or you block the cell surface integrin, the cells don't stick anymore. So thrombin is activating the beta-1 integrin, making the cells stick, and it's doing it through RAP. And why I kind of liked all this is this is a paper from a figure from an older paper by Sahai and Marshall in Nature Reviews Cancer. And it looks a little complicated, but the bottom line is that in a normal cell, if it doesn't see if it's not in the right place, i.e. it doesn't see the right integrins, it's meant to not proliferate. But in a tumor cell, if integrins get activated inadvertently, even with the inappropriate physical clues, even when the cell's in the wrong place, it can proliferate. I didn't mention it, but integrin signaling can also activate Rho downstream. But what we've shown now then is that GPCRs can get into this pathway through activating Rho and activating integrins. And we think that this is an important way in which they contribute to inadvertent proliferation in tumor cells, and proliferation in the wrong place. And just one kind of one way that we've tested this, and we have many of these things that we could, many signals in the pathway that we could look at, but here what we've done is to silence RAP1 through shRNA, selecting RAP1 knockout cells, and then doing an in vivo xenograft tumor cell assay, a flank orthotopic xenograft, it's a flank xenograft, and show that these glioblastoma cells that we have here and another line of human glioblastoma cells as well, treated the same way, don't grow tumors when RAP is knocked down. So we're doing similar things with knockdown of other components of this pathway. I told you that we said AP1 gene expression was regulated by RhoA, but it turns out we know now that the downstream transcriptional responses of Rho are probably much more selectively regulated through other transcriptional co-activators. One of them is MRTFA, which is a cofactor, it's a transcriptional co-activator for SRF. A number of people have studied that, and we published a paper on it not too long ago. Rick Newbig's lab has studied it a great deal recently. Another transcriptional co-activator shown more recently in work from Kunliang Guan's lab and stuff that we did as well with him is called YAP, and YAP is a co-activator for TEAD. And these had been totally studied independently. They'd both been shown to be regulators of this CCN1 gene, and then they were both shown to be activated by S1P or thrombin or LPA. And in a recent paper that Olivia Yu, my graduate student, published, we actually showed that they seem to work together and cooperate in regulation of CCN1 and a number of other genes. So these, we showed that S1P causes the activation of MRTFA as indexed by its nuclear localization, increased nuclear staining, and activation of the luciferase reporter gene. And the same is true with YAP. It's activated by S1P, and again in a C3 dependent way. Both of those show that, and it activates gene transcription. And the sort of surprising thing was when we looked at this gene, and we've looked at many others now, when we looked at this CCN1 gene, if we knock down YAP, we totally abolish the ability of thrombin to induce the CCN1 protein, even though MRTFA is still there. And if we knock down MRTFA, we abolish it, even though YAP is still there. So both of these transcriptional co-regulators have to be, one is not enough. One is not sufficient to induce the expression of that gene in response to thrombin or S1P. And the same is true for the proliferative response. Thrombin won't cause, this is actually S1P, thrombin S1P won't cause proliferation in the absence of YAP and won't cause proliferation in the absence of MRTFA. So a lot of these signals, like proliferative signals, seem to depend on many, many things being activated simultaneously and turned on. And we're currently doing some gene arrays and again finding that there are, with RNA-seq, using YAP and MRTFA CRISPR-Cas knockout cells, and showing that there are a number of genes that are regulated in common by these two transcription factors. And part of the reason I mention them is that at least MRTFA has already been considered as a therapeutic. This is used. So work we did, Rick Newbig actually made a number of compounds that he's published on the last couple of years, and this is these two compounds are both inhibitors of MRTFA translocation to the nucleus, and inhibitors, as shown here, of expression of this MRTFA-dependent gene. We also somehow got Andy McCammon's lab interested, I guess, in this signaling pathway, and they made some inhibitors of Rho-A, and we tested them in these glioblastoma cells, and these inhibitors that they made prevent Rho-A activation by thrombin. We've yet to test them in any functional response, but we've tested some other inhibitors, and this is just showing you one particular functional response, which happens to be S1P effect on survival, which I'm not otherwise talking about, but the inhibition of survival is by S1, the effect of S1P on survival is blocked by either this MRTFA inhibitor or inhibitors of Rho. So it's definitely a signaling pathway that can be targeted. I was going to turn on my own timer, but I didn't, but I think I'm still fine. So the other pathway that I'm going to say a few words about is inflammation. You don't have to get up. I'm not at 30 minutes. I know that, Paul. Because what time did I start? Am I? And why I want to mention this inflammation thing is there's a very interesting target of RhoA, and that is phospholipase, a novel member of the phospholipase C family, which is called phospholipase C epsilon. And phospholipase C epsilon is shown down here compared to the other members of the phospholipase C family. And what is unique about it is several things. One is this has an extended domain that contains a RAP exchange factor, and I'll tell you something else about it. But what we had discovered using knockout cells from making astrocytes from PLC epsilon knockout cells is that the ability of thrombin to stimulate PI hydrolysis in these astrocytes, in wild type and knockout astrocytes, was highly dependent on PLC epsilon. So was the effect of LPA, so was the effect of S1P. But carbachol, which we knew worked through muscarinic receptors, GQ, the regular old PLC beta, well characterized, didn't care if PLC epsilon was there or not. So these agonists I've been telling you about all along that activate Rho seem to do their PI stuff through this PLC epsilon. And how they do it is that PLC epsilon actually has a binding site for RhoA and can be turned on by RhoA binding to this binding site that Ken Harden described. And so that's how that happens. And just a couple of slides about the inflammatory response. Stephanie Dusaban, a student in my lab, had shown that thrombin, LPA, S1P all induce the expression of COX-2 and a number of other inflammatory mediators and cytokines. But this does not happen in the absence of PLC epsilon. So the inflammatory response to these Rho activators is mediated through PLC epsilon. There's a lot more data in the papers that she published, and she also determined that activation of NF-kappa B, which everyone knows is sort of a super regulator of inflammation, is also dependent on PLC epsilon. And the pathway that I won't detail is something like this, that the PLC epsilon generates diacylglycerol, activates protein kinase D and NF-kappa B, COX-2, and other inflammatory genes. And what's uniquely important about the PLC epsilon is that the DAG response, that is, it is compartmented in an intracellular Golgi-like compartment, a paranuclear compartment, and that it is activated by the RAP, it's activated in a constitutive way because of its ability, because it has this, go back Tuesday, because it has this RAP, this CDC domain, and activates RAP, and I didn't go into this, but RAP feeds back on this RA domain. So it's a way of getting sustained signaling through PLC epsilon. You're mostly making DAG. It's not really involved in IP3 and calcium signaling because of where it's localized. And we tested the importance of it in a couple of disease or in vivo models, and one is a stab wound injury model where we just did stab wound. And remember, these are astrocytes we're interested in. There's astrogliosis in the stab wound injury, and you saw it in that other picture, but you can measure it by increases in GFAP staining. And then the inflammatory response by COX-2 or the MCP1. And in all of these cases, this effect of the stab wound is on the inflammatory signals, astrogliosis, and signaling is prevented if PLC epsilon is absent. And my on this last slide is that we've tested it in another model using here the PLC epsilon knockout mice in a model of multiple sclerosis, an EAE model, and you can see here that in the absence of PLC epsilon, clinical scores are very much diminished, as is the increase in these inflammatory cytokines and INOS in the spinal cord. So in summary then, I think we started with this toxin, C3, which has allowed us to implicate ROA in a number of important responses and understand the downstream pathways should we want to interfere with them at this level. That might be possible, but also I think that GPCRs that activate not only thrombin S1P but orphans that activate this pathway, or the G12, 13 proteins and their regulation are certainly targets for therapeutics that would affect these responses in diseases and pathophysiological tumor growth, fibrosis, and inflammatory CNS disease. And so just, this is not all the people in my lab, but the ones who've worked on this project here. I won't go through and name them because now Paul's really standing here. So thank you for your attention.
Thank you, Joan. We have time for a question or two before the break. Any questions? Roger, can you shout it out?
I can shout out.
I might not be able to hear you. Run up there.
All right, you got it.
Thank you. So, Joan, it's a wonderful talk. I'm curious whether you think that the coupling is going to be cell context dependent, environment dependent.
The coupling.
The coupling to Rho directly through 1213 and specifically through thrombin mediated, for example.
Which receptors coupled to it?
Yeah, so is it, because thrombin receptor has been known to couple to many different types of G proteins.
Yeah, sure. I mean,
the one that's interesting, I think, that has to do with Rho is Q and how Q can regulate trio or
right.
P63.
Yeah, I mean, so I didn't go into it. Of course, there's different gaps and different receptors on different cells, and thrombin can activate other signaling pathways. So can S1P and LPA. We were lucky in these astrocytes happened to really sort of, maybe because in our very early stuff they had a lot of Rho, and maybe a lot of G12, that they sort of, all of those receptors tend to go that route, but certainly that they don't have to. And so, of course, one has to know that, and it will be cell-specific for sure.
Any other questions? Okay, if not, we'll take about a 17-minute break or so. We'll start again at 1:15. That'd be great. Thank you. Thanks to everyone who participated. A red car.